This video is going to introduce you to the idea behind the research that developed the concept of linked genes. So if we were to go back and reference this in a historical context, there was a gentleman named Thomas Hunt Morgan, which would be the individual who really took Mendel's idea to the next level. And in class we talked about the idea that people started to realize that traditional Mendelian probability was not able to predict outcomes in all circumstances. While in limited scenarios they were predictable and accurate, you'll see in this activity that you'll start to see where that deviates from being applicable under all scenarios. So in our scenario today, we're going to look at the possibility of genes being linked between wing shape and the color of fruit flies. And we're going to use a simulator for this so that we can do replications or iterations very quickly. And this is also the same software that you will use in your activity on Monday in trying to not only discover what genes are linked, but also map them on a chromosome. And I'll talk about that here in a little bit. So the flies that we are going to use are male and female, and the easiest way to differentiate between that is on the posterior end. Females will have a more pointed posterior end, and males will have a rounded posterior end. Males will also have comb bodies up here on their forelimbs, but being able to identify those is just easier for this activity. All right, so we are going to cross a wild type female with a mutant male, and what we mean by that is wild type is the normal occurrence of a particular trait. That's not an indication of dominant or recessive. It's just the more prominent phenotype that's seen. The goal here is to determine whether the color of the individual and the characteristics of the wings are linked, and if they are linked, to what degree. So we're going to kind of go over and look at the simulator here in a second. But we are going to use some Punnett square analysis because what we want to see here is we need to do an F1 cross, and that will allow us to determine the dominant and recessive characteristics of the traits. So we're going to go into our simulator and we're going to order flies. So the first one that we need to order is the female wild type, which is right here, and we can add to cart. The next thing we need to do is order the males. We want body color black and wing shape. This is technically called vestigial. So we want to order those. We can check out, and then they should be shipped. Hey, there they are. All right, so what we want to do is unbox these, and we want to mate these particular flies. And this will allow us to see, based on the offspring, what we will more than likely see in terms of dominant and recessive. Because if we were using standard Mendelian genetics, we should kind of be able to predict the probabilities of what the F2 generation would look like. So they're in there, there's mating, and this takes a little bit of time. So the one thing that I do want to mention while that is happening is that these original crosses are true breeding, and what that means is they have the same alleles for both traits. So if we were to write that down, we can use colors here to help indicate that. So the way that this is indicated is this would be black but wild type, and what's called vestigial, and the plus indicates wild type, so wing shape. And this one is recessive all the way down, so no pluses. And we could make a reasonable prediction here that we are going to have heterozygotes. We just don't know what the phenotypes are going to be. So if we were to cross these based on allelic combinations, we know that all of them are going to be a heterozygote. And we just need to indicate what the what that phenotype is going to be. So I believe that our cross is done, and we can open the incubator, select these. They're going to be knocked out with fly nap, which is just an ether which puts them to sleep. And then what we can do, since they're small, is look under a dissecting scope, and we can sort these. And so they're sorted by male and female in this particular characteristic. So observationally, we can see that there's similar quantities, and this can be verified. We see that we have 587 females and 598 males. So the fact that they're evenly distributed probably means that this isn't sex-linked, but we'll be able to verify that later. So if we click on the females, we can see what all of the females look like. And what we can see here is that they express yellow and regular wings. All right, so that's important to know. If it was sex-linked, we would potentially see something different in the males. We will need this individual again in our F2 cross. So what we can do is we can use in new mating, and we'll revisit that here in a minute. Whenever we zoom out, we can then check out the males. And what we see here in the males is we see yellow body type and standard wings. So if we were to back out to our cross here, we would know what we had before, that they were all heterozygotes. So BL plus, BL, VG plus, and VG is heterozygous, but what that means is the wild types, the yellow and the non-wrinkled wings or the non-vestigial wings, are dominant. So we know that the wild type characteristics are dominant to the recessives. And that's important to know moving forward, because that should help us predict probability, or what we would expect to occur with an F2 cross. So what we can do in that regard is then go back, and what we want to see is if, now that we have a heterozygote, if there's linkages between the two genes that are seen. So what we can then do is we can return to the lab, and we have a female down here that is yellow with non-vestigial wings or non-wrinkled wings. What we can do is we can go back in and we can cross that with the original male genotype, and then we can look at gene linkages in terms of probability, because we should be able to predict this. So I combine those together, and they are mating. And so if we go back here, we can kind of look at this. We know that all of our individuals now are heterozygotes. So BL plus, BL, VG plus, and VG. Where we know all these individuals are homozygous recessive. Oh, sorry, there's no plus there. All right, so the reason this is important is because we now will have a variety of genotypes that will be expressed. We won't have one uniform version of this. So what we would be able to see here is we will have, let's see, I wrote these down already so I didn't have to do them live. So let's see, we've got BL plus and BL, which is the only thing that can be donated, VG plus, and then VG is one option. We've got BL plus, BL, VG, VG. We've got BL, BL, VG plus, VG, BL, BL, VG, VG. So these are all the phenotypic probabilities that we could potentially see. Now we would see that for male and female evenly distributed if they were not linked. So that's what we're going to do now, and that's what we just crossed. So what we can do is go back to our test cross. They should be done. Open the incubator, check out cross 2, and then what we should see here is 8 evenly divided piles if Mendelian genetics were correct. So when we sort these flies with the paintbrush so they don't, die, they're just asleep, we can make an immediate observation that we do not have 8 evenly distributed piles. And this shows us an indication that there is some degree of linkage. So what we can do is we can send this data to the computer and we can analyze some things. So the first thing I want to look at is sex linkage. And we would see a difference in percentages in male and female of certain traits if we had sex linkage. So if we look at male wild type, we see 265, female wild type 239. That's a reasonably close. That's 2 percentage points. That's not that would not end up falling within parameters for a statistical difference. So we can actually ignore the sex here. So the other thing that we want to notice is looking at wild type and mutant. So we actually have some wild type and mutants here. And what that would mean is it seems like in this case those two genes are traveling together. And the reason for that is we have a very low percentage of, one version of one gene, one version of the other. It seems for the most part in almost 82% of cases the genes traveled together. Now that's really important here, because what we should have seen was equal probability, but we do not see that at all. So if we go back to our idea here, we see significant differences between the traits that we are seeing. And the reason for that is that they're linked together. So let's talk about the mechanism for that to make sure that we understand how this is happening. So linked genes means that they're traveling together. And we know that these genes are on a particular chromosome. Now, if we go all the way back to this idea of meiosis, we know that there's this phenomena called crossing over, and this is great for increasing genetic diversity. However, we know that if these are two homologous chromosomes, they will exchange genetic information in crossing over. And let's say that they exchange this part of their chromosome. So crossing over has occurred. There has been genetic exchange. But what you see here is that these genes have not been separated. So they are still traveling together. Now, this is pretty unique because we can actually tell how close genes are linked together by the percentage of observations in terms of what we see. So we saw something along the lines of 82% of them traveling together, which would mean 18% separate. This is wildly different from the 50-50% that Mendelian genetics has in play. So we can see that there is a strong linkage here. And this is important to note because these percentages are important. They could actually give us a mathematical length of difference in terms of how close they are on a particular chromosome. So what we want to notice is that if we see a high percentage of linkage together, we know that they're closer together on that particular chromosome. The closer you get to 50-50, the further away they are. And this 50% number is pretty good. If you have less than 50% of them being linked together, what that means is you do not have linked genes. If you have more than 50% linkage or them showing up together, that normally means that you have a linked gene, that you have some sort of a characteristic that is moving beyond traditional Mendelian probability. So if we were to talk about these other ones, these, what in this case was the 18%, these 18% where there is exchange is called recombinant. And that means that there was two separate genes being combined together. These are the indicators of distance. So we know that random fertilization should be 50-50, but what we want to do in terms of genetic mapping, which is where we're going, is thinking about these recombinants. These are the examples of where those genes were split apart. This holds the key for determining their location from each other. So Alfred Sturtevant, which was one of Thomas Hunt Morgan's students, predicted that the farther apart the two genes are, the higher the probability for a crossover or recombinant would be present. So if we see a higher percentage of recombinants, that means they're far apart. If we see a high percentage of linkage or a low percentage of recombinants, we know that they're really close together. And that would allow us to map where genes are on chromosomes, which ultimately allowed us to make more reasonable predictions about where things are. So when we talk about gene mapping, the distance between genes can be expressed as map units. One map unit, or centimorgan, so what you get to do when you find something out is name it after yourself, represents a 1% recombinant frequency. So if we have an 18% recombinant frequency like we saw in our cross, we know that those two genes are 18 centimorgans apart. Now, what you can do, and this is what activity we'll do on Monday, is you can look at a variety of genes, 2, 3, 4, 5, innumerable numbers of genes, and actually map them on the chromosome in order to determine where they are found on that chromosome and also if they're linked based on the percentages. So you want to make sure that you kind of review this idea of analyzing and determining recombinant percentages. We're going to use the same simulator I used to do this, but you'll be engaging in a similar activity, collecting data, and then ultimately mapping genes on a chromosome based upon their recombinant frequencies.